Cambridge, 1978. In a quiet ward at Addenbrooke's Hospital, a man on dialysis is about to receive a kidney that his own body was never built to accept. The donor is dead, the match is imperfect, and the surgeon standing over him is about to try something that has never worked in a human being before this exact night. Surgeons had already solved the harder problem, or so it seemed. They could remove a failing kidney. They could stitch a foreign one into its place, connect the vessels, watch it fill with blood, watch it sometimes start producing urine within minutes. The hands were good enough. The instruments were good enough. What refused to cooperate was something no scalpel could touch: the immune system. For decades, that had been the real obstacle in organ transplantation, not the surgery itself. The body does not know that a kidney, a liver, or a heart has arrived to save it. To the immune system, every transplanted organ looks the same. An invader gets attacked. Doctors had tried to stop that attack before. Corticosteroids, azathioprine. Whole regimens strong enough to blunt the immune response and just as often strong enough to leave a patient defenseless against the next infection walking through the door. Rejection killed patients, so did the drugs meant to prevent it. Physicians were choosing between two different ways of losing the same person. By the late 1970s, the numbers told the story better than any textbook could. In some centers, liver transplant recipients had barely a 1 in 3 chance of seeing their next birthday. Heart transplantation, which had opened the world's imagination just a decade earlier, was quietly losing momentum. Too many operations, too few survivors. Some surgical teams were starting to ask, quietly, whether transplant medicine had a future at all. And yet, on this particular night in Cambridge, something is different. The substance now entering this patient's body did not come out of a research program built to fight organ rejection. Nobody designed it for this. Its origin has almost nothing to do with transplant medicine, almost nothing to do with hospitals at all, and that is part of what makes this story worth telling. Because somewhere, years earlier, in a place with no surgeons and no patients waiting for a match, the answer to this exact problem was already sitting in the ground. Nobody knew it yet. Stories like this one rarely start with the discovery itself. They start with the waiting. The phone that might ring. The name that might be called. A body that might or might not accept what medicine is offering it. If you have ever waited for news like that, for yourself or for someone you love, you already carry a piece of this history, whether you know it or not. That is worth sitting with for a second before we go further. If this story is already pulling you in, a like helps more people find it. And this channel exists because of exactly that kind of quiet support. And if you have ever waited on news that could change everything, say so in the comments. You are not the only person watching who has. There is a lot more ground to cover here, and where this story goes next will surprise you, even if you think you already know how it ends. Subscribe if you want to be there when it does, because what happened after this night in Cambridge changed medicine in ways almost nobody expected, including the people who lived through it. Because the answer to this patient's problem was not found in a hospital. It was found by accident, years earlier, in a place nobody was looking for it. Before we go any further, I want you to sit with something for a moment, because until you feel the weight of it, nothing else in this story will land the way it should. Picture what it actually means to live on dialysis. Not for a week, not for a bad month, but for years, waiting. 3, sometimes 4 times a week, you are connected to a machine that does what your kidneys no longer can. Blood leaves your body, gets filtered, and comes back. Hours at a time, every single time. You plan your entire week around a chair in a clinic. You watch your energy fade a little more each year, even on your best days. Because a machine, however advanced, is never quite the same as an organ that was built specifically for you, inside you, before you were even born. And somewhere out there, on a list you cannot see, your name sits next to hundreds of others, waiting for a kidney that might come next month or might not come in time. You learn to carry your phone everywhere, even into the shower, even to bed, because the call that changes your life does not wait for a convenient moment. This was the reality for renal patients heading into the late 1970s. And it is worth remembering that organ transplantation itself was still a young, fragile branch of medicine. Surgeons had only been attempting it seriously for a couple of decades. Every hospital that offered it was, in some sense, still learning as it went, refining technique after technique, and quietly accepting a level of risk that would be almost unthinkable in a modern operating room. Now imagine the phone finally rings. A kidney is available. It came from someone who died, someone you will never meet, whose family made a decision in the middle of their own grief that will, quite literally, decide whether you live. The match is not perfect. It rarely was in 1978. Tissue typing was crude by today's standards, and cadaver donors rarely lined up neatly with the patients waiting for them. But it is close enough, and close enough was what medicine had to work with. You are wheeled into surgery. Surgeons connect the vessels one by one with a precision that took decades of trial and failure to perfect. The new kidney is placed, the clamps come off, and then, in that operating room, everyone watches for the same thing. Does it turn pink? Does it start to work? For a moment, in operations like this one, it does. Blood flows in, urine begins to form. Everything a surgeon can control has gone exactly right. There is a kind of relief in that room that is hard to describe unless you have felt it yourself. The quiet exhale of a team that just pulled off something the human body was never designed to allow. But that relief never lasted as long as anyone wanted it to. In the days that followed, patients were not sent home to rest and heal in peace. They were watched constantly through blood tests and biopsies and small worried conversations at the foot of the bed. Every rise in creatinine, every fever, every subtle shift in how a patient felt that morning could mean one of two very different things. And doctors would not always know which one they were looking at until it was too late to change course. Was this an infection? Or was this the first sign that the body had started fighting back? Nobody in that room could always tell the difference, not with certainty, and that uncertainty alone was enough to keep an entire surgical team awake at night. But here is the part almost nobody outside medicine understood at the time, and it is the real subject of this entire story. The surgery was never the hardest part. Connecting a kidney to a body is mechanically a solvable problem, and by the late 1970s, surgeons around the world had solved it more times than anyone could count. What they could not solve, not reliably, not yet, was something far more stubborn. Something no scalpel could reach, no matter how skilled the hands holding it. Your immune system does not know that this kidney arrived to save your life. Nobody gave it that information. All it knows is that there is tissue inside your body that does not carry your genetic signature, and its entire purpose, built over millions of years of evolution, is to attack anything that fits that description. A virus, a splinter, a transplanted organ. In that first moment, your immune system treats all three exactly the same. It does not negotiate. It does not pause to consider that this particular invader is the only thing standing between you and a return to dialysis, or worse. That is the part that should stop you. A medical team can do everything right. The surgery can be technically flawless, the match as close as anyone could find, the timing perfect. And the same body that is supposed to heal can turn around and try to destroy the very thing that was placed there to save it. If you have ever loved someone who went through something like this, or sat helpless while a decision that mattered most was completely out of your hands, you already understand a version of this feeling. The sense that you have done everything humanly possible, filled out every form, made every difficult call, and now you are simply waiting to see whether your own body will cooperate with the plan. That contradiction, a life-saving organ treated by the body as an enemy, sat at the center of transplant medicine for decades. It is easy to say those words quickly and move on, but stop and actually consider what it meant for the families living through it. Every transplant carried two separate outcomes stacked on top of each other. Did the surgery work? And days or weeks later, will the body allow it to keep working? Two different battles, and losing either one meant losing the person you had just gotten back. By the time our patient in Cambridge was wheeled into that operating room in 1978, this contradiction had already cost thousands of people their transplants, and some of them their lives. Doctors were not blind to any of this. For years, they had been trying to quiet the immune system down using whatever tools existed at the time. Powerful drugs, entire regimens, all of them capable of holding rejection back to some degree, and every single one of them came with a brutal trade-off of its own, one that put patients at the mercy of an entirely different danger. What those drugs actually did to the people who depended on them, and how far doctors were willing to go to buy their patients a little more time with a working organ, is exactly where this story needs to go next. Because if you assume the danger in that operating room ended the moment the kidney turned pink, you are about to learn just how wrong that assumption really was. If any part of what you just heard hit close to home, whether you have waited on news like this yourself or watched someone you love go through it, type the word "waiting" in the comments below. I read everyone, and I promise you, you are far from alone in that feeling. So what did doctors actually have back then to keep that invisible war under control? Not much, and what they did have came at a price most patients were never fully warned about: corticosteroids, azathioprine. That was, more or less, the entire arsenal against organ rejection for decades. These drugs did not target the immune cells attacking your new kidney specifically. They could not with the technology and the biological understanding available at the time. Instead, they suppressed your immune system broadly, indiscriminately, the same way you might silence an entire room to stop one person from talking. And a silenced immune system does not just stop attacking your transplant, it stops protecting you from everything else, too. Think about that trade for a second, because it is worth sitting with. You survived the surgery, your new kidney is working. And now you are more vulnerable to the common cold, to pneumonia, to infections that would barely slow down a healthy person but could kill you. Patients on these regimens developed fragile bones, diabetes, uncontrollable blood pressure, faces that swelled beyond recognition from the steroids alone. Some survived the rejection only to lose the fight against an infection nobody saw coming. Doctors were not choosing between danger and safety. They were choosing between one danger and another and hoping they had picked correctly. And here is what almost never gets said out loud about this era of medicine. The patients living through it were not passive. They knew on some level exactly what bargain they had made. Every fever became a question with no easy answer. Is this nothing, or is this the beginning of losing everything again? Families learned to watch for warning signs the way soldiers watch a tree line, quietly, constantly, because the alternative was not watching at all. This is the world our patient in Cambridge was living in. Not a world without options, but a world where every option carried its own quiet cost, where survival meant trading one kind of fear for another, and where medicine badly needed something it simply did not have yet, something precise enough to calm the rejection without leaving the rest of the body defenseless, something that, as far as anyone in that room knew, did not exist. Now take that same fragile balance you just felt with kidneys and multiply the stakes considerably. Because if a failing kidney was frightening, a failing liver was something else entirely. At some of the leading transplant centers in the United States going into 1980, the numbers were brutal. Roughly 1 in 3 liver transplant patients was alive after a single year. Not 5 years, not 10, 1. Surgeons had already proven, again and again, that they could remove a diseased liver and put a new one in its place with real technical skill. What they could not prove, not consistently, was that the patient walking out of that operating room would still be here to see the next spring. Heart transplantation was not faring much better. A decade earlier, the first heart transplants had made headlines around the world, filled people with something close to awe. By the late 1970s, that excitement had gone quiet. Programs were shrinking. Some surgical teams had simply stopped, not from losing their nerve, but because they could no longer justify the odds to the families sitting across from them. If you have ever sat in a waiting room doing that same quiet math, weighing hope against odds nobody wants to say out loud, you already know that feeling in your chest. Somewhere in that discouragement, a question was starting to form that almost nobody in medicine was asking yet. Now, let's step away from that operating room for a moment, because the answer to everything you just heard was not sitting in a hospital at all. It was sitting in the ground 9 years earlier, in a place about as far from a transplant ward as you can imagine. Norway, 1969. A quiet, windswept plateau called Hardangervidda. No hospitals here, no surgeons, no patients on dialysis. Just moss, rock, and soil, and a scientist on vacation who happened to work for a pharmaceutical company back in Switzerland. His employer had a habit. Every researcher who traveled was quietly encouraged to bring back a handful of soil just in case something living inside it turned out to be useful. Most samples went nowhere. Most still do, in labs everywhere. But this particular handful, scooped up almost as an afterthought, was carried back to Basel and logged into a routine screening program like 1,000 others before it. Nobody in that lab had organ transplantation on their mind. Nobody was thinking about kidneys or livers or a patient in Cambridge who had not even been born yet, medically speaking, as a case. They were hunting for something else entirely, something with a much simpler, much more marketable use. If you have ever stumbled onto the right answer while looking for something completely different, you already know how strange that feeling is, and it is exactly what was about to happen here. Drop the word accident below if you have lived that same strange coincidence. Back in Basel, that soil sample went through the same first step every sample went through. Scientists isolated the organism living inside it, a slow-growing white fungus, and ran it against a panel of standard tests. The same test they ran on hundreds of other samples that year alone. What they were hoping to find was an antibiotic. In the early 1970s, that was still the gold rush of pharmaceutical research. Soil fungi had already given the world penicillin and a whole generation of antibiotics after it, so every promising sample got screened the same way, against bacteria, against other fungi, looking for anything that could kill an infection efficiently enough to bottle and sell. This fungus did show some antimicrobial activity, just not much. Its range was narrow, its potency underwhelming compared to what was already sitting on pharmacy shelves. By the ordinary standards of the lab, it was, at best, a mild disappointment, which is usually where a compound like this quietly disappears, filed away and forgotten, one more entry in a drawer full of samples that almost worked. If you have ever poured real effort into something that came back looking like a dead end, you know that particular kind of quiet disappointment. Almost. That word is about to matter more than anyone in that lab realized, and for a while it did get shelved or came close enough to it. By 1973, the numbers around this project stopped making sense to anyone holding the budget. Turning a promising lab compound into an actual approved medicine meant years of clinical trials, mountains of regulatory paperwork, and a bill that ran into the hundreds of millions of dollars, money the company had no obvious, guaranteed way to ever recover. Because who exactly was this drug even for? Organ transplantation in the early 1970s was still a small, uncertain field. Long-term survival was poor enough that some physicians openly questioned whether it had a future at all, spending a fortune developing a drug for a market that tiny looked, on paper, like a losing bet. More than one executive at that company seriously considered walking away from this compound entirely, closing the file and moving the budget somewhere with better odds of paying off. Think about how close that decision came to erasing this entire story before it started. No Cambridge, no patient waking up with a working kidney. This compound was one signature away from never reaching a single hospital. If something you cared about ever survived a moment that close, tell me with one word below. Survived. It survived, barely, mostly because one scientist inside that company kept quietly running it through a broader battery of tests, almost out of stubbornness. And in one of those tests, something completely unrelated to antibiotics showed up. The compound was suppressing lymphocytes, selectively, not wiping out the immune system wholesale the way corticosteroids and azathioprine did, but calming down specifically the cells responsible for recognizing and attacking foreign tissue, while leaving much of the rest of the immune system intact. Nobody had gone looking for that. There was no research program built around finding it, no hypothesis chasing this exact outcome. A researcher named Jean-François Borel noticed a pattern in the data that did not fit what anyone expected an antibiotic candidate to do, And instead of dismissing it as noise, he followed it. Selective. That is the word that should stay with you here because it is the entire answer to the question this story opened with. Not a sledgehammer against the whole immune system, but something closer to a scalpel. And the scientists in that lab were only beginning to understand what they had actually found. That word, selective, eventually reached a surgeon who had spent his entire career fighting exactly the problem it seemed built to solve. Roy Calne had watched kidney transplants fail for years, not from bad surgery, but from a body that simply would not stop attacking what it had been given. When he heard about this compound and the early animal data behind it, something in him recognized the shape of an answer. Animal trials came first, cautious ones, dogs receiving kidney transplants and for once keeping them. That was enough for Calne to take an enormous professional risk. A substance discovered by accident, tested for a completely different purpose, that had never been given to a human being for this reason before, was about to be tried on a patient who had run out of other options. Which brings us back to that room in Cambridge, 1978, and the moment we started this story with. The patient on the table, the kidney that had already turned pink. And now, finally, you know exactly what was being administered to him that night, and why a surgeon was willing to bet on something this untested. If you have ever had to trust something completely new because everything familiar had already failed you, you already know exactly the position Calne and his patient were in together that night. Leave the word trust below if that feeling is familiar to you. December 1978. 7 patients, all of them on dialysis, all of them receiving kidneys from cadaver donors that did not fully match, exactly the kind of imperfect match that so often ended in failure before this. This compound, now carrying the name cyclosporine, was given to every one of them as the primary line of defense against rejection, something that had never been attempted in humans this way before. 5 of the 7 walked out of that hospital with a working kidney. 2 of them needed no steroids at all. None of the brutal regimen that had defined transplant medicine for a decade. For a field that had grown used to counting losses, these were numbers nobody had seen before. Picture what that meant inside that hospital. Patients who had spent years tethered to a dialysis machine, watching their names sit on a list, suddenly walking back into their own lives with an organ their body had actually accepted. If you have ever watched someone get a piece of their life back after years of waiting, you know there is almost nothing that compares to that particular kind of relief. That is exactly what this result put back into a field that badly needed it. But cyclosporine's story does not end with 5 good outcomes out of 7, and neither does this one. Because inside that same group of 7, cyclosporine was already showing a darker side. Doctors saw clear signs of kidney damage and liver damage in these very patients, damage caused not by rejection this time, but by the drug that was meant to prevent it in the first place. And then came the detail that still stops people cold when they hear it, even decades later. 1 of the 7 patients died, not from rejection and not from the surgery itself, but from a severe fungal infection that overwhelmed a weakened immune system. A medicine pulled from a fungus, quietly killing a patient by way of another fungus. Nobody in that hospital could have missed the cruelty in that particular coincidence. This is the moment that separates a real medical story from a fairy tale. Cyclosporine had not arrived as a clean miracle. It had arrived as a genuine trade-off, capable of saving a transplant and capable, in the wrong dose or the wrong patient, of taking a life it was supposed to protect. Doctors were now facing the exact same dilemma this story opened with, just with a new drug in their hands instead of an old one. If a solution to one problem has ever quietly created a new one in your own life, you know that feeling. Type trade-off below if you do. And that entire dilemma, once you strip away all the medical language surrounding it, comes down to one single question. A question that would end up defining the next decade of transplant medicine and honestly still shapes it today in hospitals treating patients right now. How do you weaken the immune system just enough to save a transplanted organ, without weakening it enough to kill the very patient you are trying to save? Say that sentence slowly and you start to understand why this was not a problem doctors could solve overnight. There was no dial anyone could simply turn to a safe setting. Every patient's body responded differently. Too little cyclosporine and rejection crept back in. Too much, and the kidneys, the liver, the immune defenses meant to fight off ordinary infection all began to fail instead. This is the exact tension so much of medicine lives inside, even outside transplant surgery. Enough of a treatment to help, not so much that it harms. If you have ever had to find that same narrow line with a treatment of your own, or watched a doctor search for it on behalf of someone you love, you already understand why balance became the entire mission of an entire generation of transplant physicians. As Roy Calne expanded the trial well beyond that first group of 7, eventually treating 34 patients across kidneys, pancreases, and even a couple of livers, the early promise held up in a lot of encouraging ways. But so did the danger, and it showed up in one of the most frightening forms medicine knows. 3 of those 34 patients developed lymphoma, cancer growing directly out of an immune system that had been deliberately suppressed to protect a transplanted organ. The same mechanism keeping the body from rejecting a kidney was also removing one of its natural defenses against abnormal cells turning malignant. For a moment, the entire future of this compound hung in the balance. Some physicians started asking openly whether cyclosporine was simply trading one slow disaster for another, whether the cure itself might end up being as dangerous as what it replaced. Confidence, which had been building since December 1978, cracked. That is exactly where transplant medicine stood at this point, watching something it believed in start to look shakier the closer everyone examined it. If you know that particular kind of doubt, say so below with that one word. It would have been reasonable at this point for transplant medicine to pull back from cyclosporine entirely. Instead, one surgeon in Colorado looked at the same data and reached a different conclusion. Thomas Starzl had spent nearly 2 decades trying to make liver transplantation survivable, watching patient after patient die under the old regimens, and he was not willing to walk away from a drug this promising just because it needed to be handled more carefully. His idea was simple to describe and difficult to execute. Do not use cyclosporine alone at full strength, the way the earliest trials had. Combine it with a smaller, carefully calibrated dose of prednisone instead, and use that combination to bring rejection under control without pushing the immune system all the way toward the failures that had frightened everyone. Starzl was not testing this on a low-stakes population either. Liver transplant patients, remember, were surviving barely 1 year in 3 under the old approach. There was very little room left for error, and even less patients left in the medical community for another disappointment. If you have ever bet on an approach everyone else had grown skeptical of, simply because you understood the problem better than most, you already know the position Starzl was in. He was about to find out whether his conviction was justified. 14 patients were scheduled for that trial in Colorado in the spring of 1980. 2 did not survive the surgery itself, a brutal reminder that the operation alone still carried enormous risk before cyclosporine ever had the chance to enter the picture. That left 12 patients who made it through the transplant and began Starzl's carefully balanced regimen of cyclosporine and prednisone together. 11 of those 12 were still alive 1 year later. Read that number again next to what you already know. Roughly 1 in 3 under the old approach. 11 of 12 under this one. Not a small improvement, not an incremental step forward, but the kind of shift that changes what a surgeon is willing to tell a family sitting in a waiting room. This is the moment the entire story has been building towards since that first scene in Cambridge. Not a perfect drug, not a miracle without a cost, but a genuine, measurable answer to the question this video opened with, arriving at exactly the moment transplant medicine needed proof that the risk had been worth taking. If a number like that gives you chills the way it should, this is the one statistic from this entire episode worth typing below: 11. It is the number that convinced a skeptical medical world. Word of Starzl's results spread through transplant medicine fast, the way real breakthroughs always do, hospital to hospital, conference to conference. And once liver transplantation had proof this dramatic, surgeons stopped asking whether cyclosporine belonged in transplant medicine and started asking where else it could go. Heart transplant programs, the ones quietly shrinking just a few years earlier, began reopening with new confidence. Lung transplantation, barely attempted before this, suddenly looked realistic. Pancreas transplants, once considered almost experimental theater, started producing survivors instead of case studies. Hospitals that had quietly closed their transplant units years earlier began reconsidering. Surgeons who had trained in other specialties, watching from the sidelines, started asking whether they could build a program of their own. In the span of just a few years, transplantation stopped being the rare, desperate procedure reserved for a handful of specialized centers and started becoming something closer to a real expandable field of medicine. If you have ever watched one breakthrough quietly open the door for a dozen more, that ripple effect is exactly what was happening across hospitals on 2 continents at once. But growth this fast comes with its own kind of danger, and transplant medicine learned that lesson the hard way. In the rush to adopt cyclosporine everywhere, some centers pushed doses too high, chasing the same dramatic results Starzl had achieved without fully understanding why his particular balance had worked. The lesson that emerged slowly and expensively was that cyclosporine could not simply be given. It had to be measured, watched, and adjusted almost patient by patient, week by week. Blood levels needed monitoring. Doses needed to shrink over time as a transplant stabilized, rather than staying fixed at the aggressive levels used in that first desperate trial. As physicians got better at that balance, the numbers kept improving. Kidney transplant programs using carefully monitored lower-dose regimens started reporting one-year graft survival approaching 80%, a figure that would have sounded like fiction back in that Cambridge ward in 1978. That is precisely the skill an entire generation of transplant physicians had to build from scratch, patient by patient, through trial and error. If you have ever had to learn exactly how much of something was the right amount, you know that word already. Leave it below. Dial. By November 1983, enough evidence had piled up that the FDA gave cyclosporine full approval for transplant use across the United States, 5 years after that first quiet trial in Cambridge, 3 years after Starzl's 12 liver patients rewrote what surgeons believed was possible. What had once been a single desperate compound, nearly abandoned in a filing cabinet in Basel, was now standard practice in hospitals treating kidneys, livers, hearts, and lungs alike. And somewhere in that shift, the entire question transplant medicine had been asking quietly changed shape. For decades, the honest question in every consultation room had been, can we transplant this organ at all? And will this person survive it? After cyclosporine, after Calne, after Starzl's 12 patients, that question started sounding almost outdated. The new question, the one that still shapes transplant medicine today, was simply this: who should receive this organ? Not whether the science could work, but who needed it most, who could benefit most. Decisions that entire ethics committees now exist to weigh, all because a substance discovered by accident in a Norwegian soil sample finally made survival the expectation instead of the exception. If any part of this story changed how you think about the medicine happening in hospitals around you right now, comment the word "changed" below. There is a reason this one drug still shapes the field decades later. There is something quietly poetic about the fact that this entire story starts and ends with the same word: receive. A patient in Cambridge received a kidney his own body was never designed to accept. Doctors around the world received a compound nobody had gone looking for in the first place. And decades later, millions of transplant recipients have received a second chance that, without one soil sample from a windswept plateau in Norway, might never have existed at all. If any part of this story stayed with you, if you made it all the way here because something about waiting, about trust, about a body fighting the very thing sent to save it, spoke to something in your own life, I want you to do something small before you go. Type the words "I receive" in the comments below. Not because this channel needs a number, but because it exists for people exactly like you, people willing to sit with a hard human story until the very end, and I want to know you are out there. If this video taught you something today you did not expect to learn, tap the like button. It costs you nothing, and it tells this channel to keep finding stories like this one and telling them properly. And if you have not subscribed yet, do it now. While this feeling is still fresh, while whatever kept you watching this far is still sitting with you. This channel exists for exactly this moment, the one where history stops being a list of dates and starts feeling like something that happened to real people, people not so different from you. There is another story like this one waiting for you right now on the video already on your screen. Go watch it next.